Oklahoma City, as you may know, is a leading research hub in the nation for this particular field of scientific analysis.
So a lot of people are curious whenever we talk about sewage surveillance is, what is this thing? Well, when we think about our waste, we can in fact excrete COVID both through our mucus, which we're familiar with through the tests that go in our noses, but also in the waste that we excrete through the toilet. Our team can then go there with our sampling tools, our pumping systems, and extract samples from the sewage. From there, we use similar tests to, actually the same tests, to what folks use for that nasopharyngeal swab that we're also familiar with to determine the levels of COVID in the wastewater. This is important because it allows us to have a scale where we can, in one sample, measure COVID in the wastewater, measure COVID prevalence or infection rates at the population level from 40,000 people to 440,000 people. Sewage surveillance has many benefits. The first is that it's timely. We can detect outbreaks before they are reported. It can serve as an early warning system for COVID. Also have the ability to have geographical and functional representation with the data. And it's not biased by individual testing environments. So things like folks' access to tests or asymptomatic cases, they don't influence whether or not sewage surveillance is detecting COVID, because poop doesn't lie.
So if we had a particular part of town that we just now have a new strain in that seems to be a little more prolific, we may try to focus our vaccine efforts in that specific location. So this is very, very usable technology. And these guys won't say it, but they're actually leading in the country on this.
Instead of individual samples being sequenced, we're sequencing a whole bunch of representative viruses from a large population. When we start sequencing now, it also allows us to detect the presence of new variants in these mixtures of viruses from the sewers. It allows us to monitor the virus evolution. Mutations that have not yet been defined, we haven't studied them, we don't know what that variant is. We can actually get the signal for that long before that work's done.